issue is our school started, right? The fall semester
Yeah,
started. So
exactly.
my plan was to do this your presentation and another group member,
Hmm.
Monica. So she's supposed to give a presentations on microbial portion of the
Okay.
molecular analysis.
Yeah,
But
yeah,
I think she also got busy, so
yeah,
we could
yeah,
not do that. And also, after finishing this session, I will
yeah.
upload the recorded video and then I will start my job, right? So that's why I wanted to.
So you have a lot of things to do at the same time.
Yeah, yeah, that's right.
Okay.
Okay.
And this is already recording this session?
Um, yes, I think it started recording. So
Okay.
whenever you are ready, you can start. Okay.
So can you see my screen?
Yes, I can see your screen.
Okay. So I kept it actually short and to the point and what was important, I thought. But if there are any other questions, then they can definitely text me or through the WhatsApp group or Facebook group, they can contact me. Yeah.
Yeah,
Okay.
yeah, sure.
So today I would be talking about molecular diagnosis of influenza virus. I mean, in particular, the quantitative real-time PCR of this influenza virus, because I was working with this virus for some time in a WHO Center for Influenza Viruses, and I will just share my experience and what they have been doing there. Yeah. So in general, influenza virus, so this virus itself, it belongs to orthoviral family and it is divided into A, B, and C. So the influenza virus A and B are basically responsible for epidemics of respiratory diseases. It also has C, but that is not that mm, dangerous. And as you can see in the right, that it's a micrograph of influenza virus A. So it has very specific structure, which I will be talking about in the next slide, and that is, just a second. Yeah. So this virus, it has certain characteristics, and one of them is a negative stranded RNA virus with segmented genome. So segmented genome means it has 8 genes, which will code for 10 different proteins. And these genes are important for us because the primers and probes will be for molecular detection will be designed for these genes. So this is the protein, hemagglutinin, surface proteins, neuraminidase proteins. These are important. So the spikes that you can see here, the green, and also these purplish structures. So these two are very specific to this virus. The key I have also written it here. So all the keys of these proteins that of the genes are already stated here. So the matrix protein, M2 protein, and S1 and S2, polymerase 1, polymerase A, polymerase B1, polymerase B2. So what is important about this human influenza virus is that it has antigenic change. It means it can change its genetic information quite easily. And the two phenomena that is happening here, it is antigenic drift and antigenic shift. So the antigenic drift, it is a gradual and continuous process through which these HA and NA proteins are accumulating mutations over time during the replication. But antigenic shift is kind of a sudden sudden and dramatic change in these proteins. So, and that is also not foreseeable. So the antigenic drift kind of, one would know that with every replication or with viral replications, there will be the accumulation of genetic point mutations. But with antigenic drift, it depends on several factors, and yeah, that can be that influenza type 1A emerges in so it's a combination of two different, for example, from two different hosts. and then there's a recombination, and that could lead to antigenic shift. And that would also mean that if there is an antigenic shift, that means a new virus is produced or created, and that means that that population, we are not immune to that specific virus. So that is a very important feature of this virus. I will cut short to what we would be doing for molecular diagnosis of this gene, of this virus. So the first thing would be the sample collections. I don't go in detail of it, but just the graphic that you can see that it has to be through a nasal swab. It can be through different other processes as well, but nasal swab is the best one with a cotton. And the collection of the sample, also transportation of the sample is very important. So it should be in viral transport medium, and that should be kept at a very low temperature, and should be either, if you want to use the sample immediately, that's the perfect, but most of the time in labs you need to wait for other samples to arrive and then use the batch. So the best would be to store it at minus 20 for short time, but for long time, then maybe minus 80 or even liquid nitrogen. So that is what was recommended. So real-time PCR assay preparation. So it was because it's an RNA virus, so we need to, I did not mention any of the kits here, but if anyone would like to know the specific kits which were used during this processes, then I can also share those information. So the RNA of these specific specimens were isolated using specific kits, but several precautions had to be taken here because it's an RNA virus, and as you know, RNases, so the enzyme RNase, is present mostly everywhere, and the degradation of this of the sample is very much likely, and also the contamination of the lab personnel. So there are two things that need to be taken into care also, of course, during sample collection. And also while doing that was very important because the quality of RNA isolated at the end is very important, and it also changes the result of the PCR. That would be the subsequent step. If the kit you are using and it has an ethanol evaporation at the end, that one should make sure that it is completely done, because presence of even minor amounts of ethanol in the sample that can inhibit PCR. So the whole process would be for nothing. That's why it's very important that one should pay attention while doing these procedures. The next step, after having a good quality PC in viral RNA, would be to set up a PCR master mix. Most of the, so we also used the recommended kits by WHO, because in order to keep everything standardized with other centers. I will just share what steps we used and what were the ingredients and why those ingredients were used, because just to go in, let's say, deep down why we used each of them. Yeah. So we used one-step real-time reverse transcriptase PCR for H1 gene of H1N1, which was the virus which caused the pandemic in 2009. And the gene, as you can see here on my screen, so the gene that we are looking, so the primers and probes which were designed was for HA gene. So the primer was designed specifically for that, but what's very important is the probe. So in this type of experiment, it's called TaqMan. I will explain it in the next slide in a bit detail. if you have any question, you can also ask in between if something is not clear, or I can then, or write it down, then I can also answer it afterwards. And so this TaqMan probe, that is very target specific. Only when the specific gene target is amplified, you would be able to get a signal, so a fluorescence signal in here, what I mean. And what is this probe? So the probe is designed for a segment of DNA that is of our interest within this gene. So it will be our forward primer, then the probe, and then the reverse primer. And probe has certain characteristics. One of the important one is that it has at its 5' end, it has a reporter protein. So it has a fluorescence reporter protein. In here it was FAM, carboxyfluorescein. And at its 3' end, it has quencher. So it means when probe is intact, that means the this piece of DNA, it has at its 5' end, it has reporter protein, and in its 3' end, it has quencher. There will not be any fluorescence, which I will speak about later. But these three are the important sequences that one would know, and these are the standard sequences that were recommended by WHO. The next one was that, yeah, of course, so the master mix, so the concentration of the master mix was important. The forward primer, the reverse primer, that is these above ones, and the concentration of the probe. Some of the times there would be in the master mix, the reverse polymerase or Taq polymerase were added, and some of the times it was not added. So depending on which kind of master mix you're using, then one should use it. But since it is a reverse transcriptase PCR, then that's also very important, and one should have a look if the master mix has a reverse transcriptase protein. Which would convert the RNA, the viral RNA, into cDNA, so the complementary DNA here. Yeah, so the assay, the TaqMan that I was talking about, as you can see on my screen, I hope you can see the cursor moving. So the forward primer is designed for 5' end, and then the reverse primers are designed, and this is the probe. So it has as its 5' primer, the probe. It has a fluorescence reporter protein, and at its 3' end, it has a minor growth binding protein. So that is basically a quencher, and they are in close vicinity. So the concept of fluorescence production is basically spread. If some of you would have heard about it, that these fluorescence protein would be excited by certain wavelength, and that certain wavelength will excite this protein, and it produce fluorescence. But since the quencher is in close vicinity, and this fluorescence would excite the quencher, but quencher is basically a non-fluorescent protein, so it just absorbs the light, but it does not produce any fluorescence. So that's why when it is intact, there will not be any fluorescence produced. But as you can see in the second step, so after denaturation, so the steps of PCR, here we already have the cDNA template, right? Reverse transcriptase and then cDNA template. After denaturation and annealing of the forward primer, so what will happen here is that forward primer will anneal, and it will be extended, yeah, polymerized using Taq polymerase. Taq polymerase has a very specific characteristic, and that is that it has exonuclease activity. So that means the probe is here, it is hybridized, and the Taq polymerase, the exonuclease activity of Taq polymerase will cleave this probe. And by cleaving this probe, that means the fluorescence protein and the quencher are separated from each other. In that case, the fluorescence protein when it is exposed to the specific excitation wavelength, then it will give us fluorescence, and this fluorescence is proportional to the amount of DNA amplified. So with each strand of DNA amplified, we will have more and more of fluorescence signals. So these were the specifics of the steps. So the reverse transcription, because it was a virus DNA, so the viral sample was treated with at 50 degrees Celsius for 30 minutes. So in different protocols, it also differs, but that is the standard that was being used there. And at 95 degrees Celsius for 2 minutes, that would be to activate Taq polymerase, and also so the reverse transcriptase, it cannot function at 95 degrees Celsius, and it's a protein, so it will denature as well. And afterwards, the normal PCR, so it is at 55 degree was the optimum temperature for binding of these primers, and it was 45. It can also vary between 40 to 45. That's also important to keep in mind. After the last stage, so what is also important here are the controls, which I would like to talk about. So our each well, because it is a multiplex real-time PCR, so each well has it will give us a fluorescence for the target detection. It will also, we also have added a dye, which is ROX. So it is a basically inert fluorescent dye. It does not interact with any components of the PCR, so it does not interact with PCR amplification. It does not mean that PCR amplification has happened or not, but it just lets you know that the technicality of it, yeah. So the reproducibility of it. So what does it tell us is that anything that, for example, bubbles in the well, evaporation, condensation, or droplets, instruments, for example, if there is a electric surge or something like that, then you would be able to see with the reading of ROX. Yeah. So it is also used as a passive reference dye that enables the normalization of qPCR. There are different types, like there are different ways to get for example, viral load, if you want to say, or quantify the amount of viral RNA that was loaded in the PCR. So in the, and if you use ROX, so it will be used as a passive reference. There are certain formulas and also softwares that are already in place. So you just need to click it. You don't have to use this dye, but it was highly recommended to use it. But if you use it to calculate the normalized value, the reported dye signal, for example, in our case, FAM, is divided by the ROX reference signal. And as long as these two, so this ratio remains constant, that means that everything is fine, yeah? So the amplification has worked. And there are not these technical issues in the during the amplification. The other important thing is that we also need to add to have an internal reference genes, because it is quantification. So you need to have an internal reference genes, and what is important here is, as you can see, is that the gene should not be, the expression of this gene should not be changed over time, and that is why we will take it as an internal reference gene. There are different internal reference genes that can be used. So in our case, we used glycerol phosphate dehydrogenase in some experiments, and we have also tried RNA-P and some other, and we got better results with ribonuclease, because the expression was very stable, at least with our samples. And so while doing these experiments, that's important to keep all these factors in mind that these need to be taken care of. So when you have your, and everything, so at the end, when you have your results, so you have your CT value, so that is the fluorescent signal that you see after 45 cycles of PCR is completed. And of course, you need to see that the specimen is negative if so we also have negative controls and we have positive controls. So that are understood that need to be there. Once all these controls are there, then and we have the results, so how we are gonna interpret it. The specimen is negative when the value of CT is undetectable. That means that there is no amplification of the target DNA. That is evident. So you will not see any value and also any fluorescence. The specimen is positive if the CT value is less than 38 or 38. So that is very important because it also depends on different labs, what is their cutoff value. So in some of the labs I work, anything after 35 was suspicious and one had to repeat it. But in this specific lab, the CT value of 38 or below was a clear positive. For example, in case you have a sample which has a CT value of 38, and if you remember, we had 45 cycles, yeah? So if the CT value of that specific sample is 38 or more, is more than 38 basically, then what is recommended is that either you can use the same extracted viral sample and run it, or extract a new set of a new batch of viral RNA and then rerun it to be sure, because you are talking about a positive sample and you need to be sure if it is really positive. Yeah. The criteria for quality control, I already talked about them, but we can also, yeah, so result of the negative control should be negative. So it's a PCR, there is no exception. You need to know that it is. And the CT value of positive control should not be more than. So the positive control will also be provided along with the kit. But what we did is that we also extracted positive samples. So we had in our culture collection, we had positive samples, and we also extracted them. And in those cases, it should not be more than 28. That means if it is more than 28, then we have to rerun the whole set. Yeah. I also would like to talk about the quantifications, or maybe we can also, I just touch upon it here. So the quantification, we have two different basically ways: the absolute quantification using standard curve method and relative quantification. So in relative quantification also, you will use standard curve method or comparative CT method. In most of the labs, there are software which are available for absolute quantifications, but you need to do a calibration for having a standard curve. So how would you make a standard curve? Would be to have a specific concentration of your positive control, and then you do a serial dilution of this positive control and run it. Then you would know that if and then you plot that against the concentration against the CT value, and then you would get your standard curve. We can also talk about it in detail, but since it's most of the time done automatically, so I have not explained it here for the time purposes. But do let me know if you want to know more about it. We were doing absolute quantification in that case. Yeah, with that, I would like to thank you for your attention. Yeah, please let me know if you have any questions, and if most of you are not here, then we can also talk about them on messages or something like that.
Uh, Thank you so much. It was really nice and in depth, I think, regarding this topic, particularly influenza virus, as you talked. Mm, I have some questions. We can discuss that first. Then I think another person just joined. I think if
Sure.
he has any questions, he can also either unmute or like write in the chat, whatever.
Yeah. Yeah.
Um, so at the beginning, I wanted to like you talked about two gene, HA and NA.
Yeah.
So but for our this PCR test, like the real-time PCR that we are doing here,
Mm-hmm.
we are targeting the HA gene only.
Yes. I mean, we can also have, for example, we can also do it for HA and NA. That's also possible. It
Okay, okay.
depends which kind of, so here, if you're doing duplexing, multiplexing,
Mm-hmm.
it depends which kind of PCR, so real-time PCR you're using. If you can use more than two different fluorescence channels, then definitely you can, but there are practices that use HA and NA. Yeah.
Okay, so if I can use I can target both of the genes. In that case, I have to. I think it was my second question. Like you talked about the multiplex, but for multiplex, we know we will add more than like one or two genes. So, like our target will be more than one gene, something like that.
Yes, exactly.
So if we target HA and NA, then according to that, we're gonna design the primer and but probe will be only one, like one probe.
Okay, so probe would be for each target. If you are going to have two genes of interest that you want to amplify, right? For
Mm-hmm,
example, HA and NA, then you
mm-hmm.
also would have two probes.
Got that.
Yeah.
And I can design the same way I designed it like here. Like I can put fluorescence and I can put MGB quencher
Yeah,
and when it's gonna break down, then it's gonna produce the fluorescence as normal
exactly. So
cases.
what you need to keep in mind while design, so most of these there are a lot of literature about which kind of fluorescence protein and quencher can be used together. But you need to keep in mind several things. So the wavelength, excitation and emission wavelength of the fluorescent dye, the excitation wavelength of the quencher, and also if you have used, for example, any other internal controls for them, you will also have fluorescence, right? So you need to keep everything in mind while using reported protein for designing. Yeah.
Got that. Regarding that, I had my next question was you talked about internal reference genes
Yes.
like beta ACTB, GAPTH, or RNSP. RNSP is the most common, like
Yes.
human housekeeping gene we know
Yeah.
that normally used as housekeeping internal control and ribonuclease. My question is, all four will be our reference gene, or we can use any of that?
I would say, I mean, these are different. I just wanted to say there are different reference
Hmm.
genes that one can use,
Genes.
but it's enough to use only one.
Got that. And next question was regarding control. So normally we know we use positive control, negative control, and anti-C.
Yeah.
And another thing we can use is like internal control, meaning like I think you talked about something rocks die. Is it kind of like is it a internal control or external control?
So ROX is basically, it is an inert dye. So it is not a control for the amplification. Yeah. It is a fluorescence dye which gives you a signal only when everything is fine there, you know.
Hmm, got that.
So
So,
it is not, I mean, I have worked in other labs as well, and they don't use it very routinely, yeah. But
Mhm,
because it was a reference lab where I was working, and it had to be very precise, so that's why we use rocks. And
mhm,
then that means each well that you get a signal that
mhm,
is already normalized. You know that in that specific well, the PCR has worked. There were no bubbles in the well. There was no evaporation, or to the certain extent that was detectable, condensation or droplet. So these things were, the technicalities.
Got that, got that.
But
Uh,
in most of the lab, they don't use it. That I would agree. It was very specific. I just wanted that people here should know that that can also.
yeah. Yeah, because this concept is for me it's a little bit new
Yeah,
because the way you describe the ratio should be similar. It's kind of
it
interesting.
should be
So
constant. It
constant.
should be
Yes.
constant. Yes.
Yeah. So if the ratio of rocks and the all other like controls stays similar, then we can understand, okay, the run is perfectly done.
Exactly.
Uh,
So the what I mean, the ratio between rocks and the reported dye. Yeah. So in that case, FAM.
Rocks and reported dye. Yes, FAM. For
Yeah.
FAM and rocks, if it is constant,
Constant,
uh,
then everything has worked. There was an amplification, and the PCR technicalities were fine.
got that. And normally, what we know is like, suppose for some of the cases, if CT value is under, as you said, 38, we know
Yeah.
it's positive.
Yeah.
The lower the CT value means it got find more of that kind of genes for amplifications.
Can you please repeat your question? I didn't get it. The
Like
CT
if
values?
like suppose I got the CT value for sample A, I got CT value 20.
Yeah.
Sample B, I got CT value 25.
Yeah.
Meaning sample A has a lot of more genes than sample B because CT value is lower
Yeah.
than sample B, right?
You mean, so in CT value 20, that means the amount of viral RNA was higher. So
Mm-hmm,
therefore, the amplification was shown up above the reference that we put
mm-hmm,
there in
mm-hmm,
the earlier cycles.
mm-hmm.
And CT value 25, that means the viral load. So not specifically the, yeah, the viral load. That's very important. That the amount of viral RNA that we use.
Viral
Yeah,
RNA.
exactly. So
Uh,
that was relatively lower than sample A.
In that case, like I think one of the like at the end of one of the slide you showed, if the CT value is more than 38, we need to repeat. Can you please explain like if it is our threshold value is like 38,
Yeah.
then it should give us the negative result, right?
Yeah. Okay. So what I mean here, this is for a reference lab that I'm talking about. Yeah. I have also used gene expression analysis where we use CT value, and anything below 40 was positive and everything above 40 was negative. Yeah. So we put this standard. But these standards are the accepted ones, and I could not change them. So if we would have any specimen that had a viral that would give us a CT value above 38, just to be sure, we have to repeat it.
Got that. Got
Yeah,
that.
because the further steps of these ones was, it was not just that will stop here. So then they would go for culturing. Yeah, then they would go for vaccine production. So it was a whole package. We could not just ignore one. That's.
Got that. Got that. Because of what I like my thinking point is from like clinical perspective. That's why
Yeah.
I was thinking, yeah, you are right. Like for that kind of conditions, it should be like more precise and they need to do that again.
Yeah.
Yeah.
But it
Um,
also depends on every lab, no? So in every
mhm,
lab, you have your references, you have your standards, and there are certain standards which are agreed upon, right? The
mhm,
scientist would know that, okay, anything below that is negative, or above that is negative, yeah?
mhm.
So that is also, but here, that was a very specific case.
Got that. Um, I have another like specific questions. In the meantime, I can see another person, two-person joins. If you guys have any questions, please unmute and you can ask, or you can just write in the chat. Um, in the meantime, I can ask another questions. That is, I think at the beginning you shared with me you wanted to share some of the knowledge about quantitative and qualitative testing.
Yeah.
That's why I prepared a little bit of that. I was checking some of the information on that. Um, if you don't mind, can I ask one question on that regards, because I
Yeah,
think
sure. I mean, I didn't include it here because of the time
time
limitations that
limit.
we
Yeah,
had.
yeah, yeah. I can
And
understand.
I just wanted that we do in-depth discussion of what we already talked about. But
Yeah.
yeah, please go ahead. I will try to answer it if I know them.
Yeah, yeah, sure. So my question is like for I only spoke like some of the people who already took the SMB exams, because
Yeah,
as our target is SMB, so
yeah,
um, they said some of the cases this kind of topics is covered, like in what kind of situation we will
hmm,
need quantitative testing. Like for qualitative testing, for example, this kind of viral, it can just say, okay, detected, not detected, or like positive or negative, something like that, right?
yeah,
And quantitative, it will give me some value.
yeah,
So in terms of molecular
yeah.
clinical labs, what kind of pathogens we need to consider for quantitative testing. If you have any experience, um, uh, my question is like in terms of like suppose for mostly what we do is in molecular lab is like the PCR,
Yeah,
that's very common, and
yeah,
then the other cases we do sequencing. So
yeah.
my question is like how it will do the quantitative things, like CT value is a quantitative thing.
Yeah, so I mean, CT value is the basis of the quantitative. Yeah.
Mm-hmm.
But as I already mentioned it, kind of superficially, that
Mhm,
you can do, so there are two different methods of quantification. So it's an absolute quantification. So you
mhm,
would have a positive standard, and then you will run those positive standard, for example, in 6 different concentrations,
mhm,
yeah, with lowering concentration. For example, if you start with 10,000, then it will be 1,000, it will be 100, it will be 10, it will be 1, it will be 0.1, or something like that. That
mhm,
will be your standard curve. And from your standard curve, then you would be able to um, calculate the absolute quantity of the specimen.
mhm. Got
Okay.
that. Got that.
But there are, I mean, so these are already inside the softwares most of the labs are using. But for scientists, we had to use, for example, comparative CT value methods when we were doing gene expression analysis. So there are different ways, so relative quantification and absolute quantification. It depends why you want to do that. For example, absolute quantification would be needed if one would like to know what is the viral load in that patient from clinical perspective, for example. Yeah, if the patient is given certain treatment, certain medications, and after a period of time you would like to know if the um, if the viral load has decreased or not, if the patient is responding to the treatment or not. So that is where you would like to know specifically. That's what I would think. But I'm
Hmm.
not a clinician, so um, we were doing quantifications because if they were under certain treatments, then we wanted to know if the treatment was answering or not.
Got that. Like different kind of pharmaceutical products or medication. Yeah,
Yeah,
it can be used to see the viral load, like increasing, decreasing, or what kind of stage it is responding.
yeah,
Got that. I think at the beginning you talked about two topics. Like I think it's a very common things, but still like for some of the viewers, like I know one of the person just joined a little bit late. If you can tell us a little bit again about the drift and shift, like antigenic drift and antigenic shift. If
yeah.
you,
So the
I think you said like shift will be sudden change.
exactly.
So
So there are the antigenic change, and this hallmark of human influenza virus is. they are RNA viruses, so we know that mutations are easily happening there, right?
Mm-hmm.
And so these are called antigenic changes, and these antigenic changes occurs in two different ways in influenza viruses. So one is antigenic drift.
Mm-hmm.
And antigenic drift means that um, with passage of time, with each viral replication, there will be point mutations which will be accumulated in HA and NA genes. That's also one of the reasons that with time we need to get vaccinated every year, because when the antigens changes, right, that means the vaccines are not answering
Mm.
specifically anymore. That's one of the reasons that for RNA viruses you need to get vaccinated repeatedly.
Mm.
Okay, and then we have antigenic shift. So antigenic shift is that it is a more dramatic and abrupt type of change. So
Mm.
it means an H1 and N1 genes. And how does it occur? That is very important. There are different ways that it can occur, but it would be, for example, that the H1 gene of one
Mm-hmm.
subtype and N1 of another subtype combines together. So then it is a new influenza virus type.
Mm. The change will, yeah, got that.
Exactly. And then we would have to design new vaccines for them. So, for example, if you see there are H1N5, yeah,
Mm-hmm.
so because there's H1N1,
Mm-hmm.
right? But there is
Yeah.
H1N5
Mm-hmm.
because N was different. H was the same, but N was different. You know what I
Mm.
mean?
Yeah,
So, uh, yeah, so these things are important to keep in mind, I think.
got that. I think then the most common question gonna come is like for I think I heard most of the people ask for COVID, it will be like this kind of situations as you said, like the like for influenza virus, like flu shot we take every year.
Yeah.
It will be common for COVID also.
I mean, you have seen that COVID has mutated itself quite a lot from the beginning of 2000 or end of 2019 till now, right?
Mm-hmm.
And that is
Yeah.
the reason that they are changing. And we have to do these booster shots. We
Mm.
also need to ask ourselves, why do we need booster shots?
Mm.
Yeah.
I think just recently in USA they passed the thing like from this fall they're gonna start another booster shot. We already got three shot, so
Yeah.
it will be like fourth shot. I think we have to live with this thing
Yes.
forever. Okay. So thank you so much, Dr. Amnam. In the meantime, Ahmed or Sana, if you have, do you guys have any questions? You can unmute and ask, or you can just write in the chat. Um, if not, thank you so much, Dr. Amnam. And I just wanted to inform you, like some of the people shared, like for SMP, biostatistical knowledge is also important. So I will like okay, Sana, don't worry. I will share the video and you can just look into that. And if you still have questions, you can just message Dr. Amna. She is in our group. You can write in the group also. I think she can answer, and everyone can be able to also see this. And yeah, so I will share some of my. I just finished the biostat class. Like there has some of the topics like confidence interval, the some of the other statistical topics. People said it's important. Also, like for as it's kind of like directorial level things, they ask a little bit of tough questions,
Yeah.
like cost analysis and clinical lab. Management kind of things. So
Mm.
for from my part, I will share some of my notes in Biostat, and if you guys have any questions, I can discuss or like we can look into that, and you can ask questions. Because I what I realized is like a lot of people, those who has some of the people has exam in this October and November,
Yeah.
and They say they finished their like a bachelor's master's long back, like 5 years, 10 years
And then you
ago.
forget it, of course, if you don't
Yeah,
understood.
yeah. So I think it's just better to discuss and remember that kind of topics again. So I think it will be helpful for everyone. So I will share. You don't have to look into all everything. I will try to share only the topics that people say it is very important, and we need to have understanding of that,
Yeah.
because for confidence interval the formula, I just finished the course, it's still like I forgot, because remembering this kind of formula is very
Yeah, I
difficult,
mean,
right?
of course, yeah, yeah.
Yeah.
But thank you so much. Actually, that would be very helpful just to know that what
uh,
material we need to go through. But because I am coming from research side of this molecular biology, so we kind of have to deal with biostatistics
yes,
every second day, because
yes, that's
the
right.
data needs to be analyzed and it has to be published and things like that. But
Mhm.
definitely it will be a great asset if you share it on our group and then everyone could have a look at it. If
Mhm.
there is anything that I would be able to help with, I can do that as well, because I have the practical experience of using those formulas in different studies that we are conducting. So
Mhm.
then do let me know if there is anything I would try to. Yeah,
Okay, yeah, sure, sure, that sounds good. Thank you so much for joining. I'm so sorry I took it this little bit early morning, because I have to just get back work, and hopefully you guys will watch this video maybe later. Thank you so much, Dr. Amna, for
you're
giving
welcome.
me your valuable time, and
You're
have a
welcome.
nice day,
Yeah,
everyone.
thank you. Thank you, everyone. Take care. Bye.
Bye bye.